Our next speaker is Daniel Robertson from Eli Lilly. He's going to talk about the Open Innovation Drug Discovery Program that they have going on.
First, thanks for the opportunity to come and speak to you today. I mean, when Alex reached out to me and said, hey, can you present? I was saying, well, we presented S prime, which is one of our other key tools that use ChemAxon, and we're not really there yet at Lilly relative to what you've seen some of the other pharmaceuticals in terms of replacing our legacy systems. But there is one program where we very much use the ChemAxon tools as we've moved forward in the open innovation area. And so what you see is we're using ChemAxon tools really in a couple of our key innovative areas, and then hopefully at some point in the future you'll see me be able to talk about where we'd leverage it in other areas of the organization. And so I've been involved with the open innovation effort here at Lilly from when it started, from back when I was actually in the research side of LRL. And when you actually ask what's open innovation, it really comes back to, as we go forward in the pharmaceutical, we're looking for new ways to find how do we move forward and innovate. You've seen all the diagrams. It seems like we're spending more money not getting the output that we want going forward. So in terms of the open innovations, we think about is we can't continue to rely on our current research. We need to shift the paradigm to use external ideas, external data as they may be, and really blur that boundary between the firm and the environment to allow those to go back, but still in a way that most people benefit from the engagement. That's not a one-way street. So Lilly has as its part for the philosophy, like most of the pharmaceuticals, really innovative therapies, partnering collaboration. We've had that for a lot of our key products that we've delivered, successful partnerships, and then at the same time continuing to develop our staff and look for innovation as we go forward in the future. And in fact, if you walk the hallways, it really has a key focus on innovation as we go forward. And so if I just do a quick overview here, don't want to get into too much debates in terms of these. Actually, John gave a good intro to a few of these things. Really, at Lilly, we look at the whole therapeutic or the whole spectrum of ways that you can interface and modify disease, all the way from small molecule, which is more of my background, up through antibodies, biologics, and some of the newer bispecific antibodies, or again, those things that are going for various planned polypharmacology or multiple therapeutic at the same time. And if you look at Lilly and our portfolio at this point, Lilly's started off, one of our first collaborations was actually in insulin with the University of Toronto. So Lilly's been in the biologics for a long time. And when I first joined about 2000, the mix was probably under 40% biologics in our late stage. And now we're actually more at a 46% is what I saw recently, biologics, 54 small molecule, and you'll expect maybe that to change for Lilly as we go forward to be a balanced small molecule biologics company going forward. Where I'm going to focus on a little bit on the small molecule side, because that's where we started with the open innovation effort. And so if you look at this in terms of going, I'm not going to go into debating around here. Lots of people internal to Lilly will call phenotypic drug discovery the neoclassical, meaning let's go back to how we found aspirin or all those other type things in terms of let's do a phenotype we want, and we don't care what the mechanism of action is or the target in terms of that is. It still remains to be debated if we can still get regulatory approval for those where we haven't defined the targets, at least in the new case as we go forward. And then also as John talked about, a key premise has been really the target-directed drug discovery as we go forward. And actually with the we had continued to have an effort on fragment-based approaches, trying to build molecules up. And when we acquired SGX Pharmaceuticals in 2008, it gave us actually more structurally enabled capabilities to be able to do that. The whole goal, though, on this is in the end state, especially if you're going targeted based, is at some point you've got to get it into the organism, human, and have it validated that it really is productive therapeutic. We've gotten a lot better in terms of understanding toxicology, those type things. Most of our failures now are that they're failing in phase 2 to show differentiated performance in terms of this, in terms of actually interacting with the disease state. So if I sort of move forward with where we were at, was in the 2000s, we did a huge effort, like many of the other individuals, companies, in terms of improving our compound collection through acquisition of what was available on the market, through what was planned small libraries going after targeted areas. And the head of our discovery chemistry actually over the holidays actually was coming back and saying, where do we go next? Have we reached the limit of our compound collection? What are we going to do, and what's the challenges? And are there ways to access other innovation or compounds? And it came back to saying, couldn't we reach out and do academia and small biotech? Lots of times we'll reach out and do collaborations, but can we do it a different way, where we bring something to the table that's useful for the academicians, and then we get an opportunity to see what innovation is out there and potentially go to collaboration. So this was an opportunity for open innovation. So a goal was to expand discovery organization to access around the globe. We didn't care. Whoever wanted to participate. We do it unbiased, and what I mean by unbiased is that we would actually use our assays, the same things that our scientists are using to do their SAR, and be able to qualify and quantify how those external entities actually respond in the same things that our project scientists use. And then also the goal is not to just acquire a compound, because you can analyze the compound, but in discovery side, there's still a huge amount of SAR you have to do, a huge amount of work to actually get to a clinical candidate before you even go to human dose. So we actually wanted to access the potential of the individuals, the ideas behind it, them to drive the SAR. And then in the end, we wanted to do this with minimal cost, if we could, leveraging the infrastructure we had in place and moving forward. And so as we moved forward in this, not a trivial task, we actually did launch in September of 2009, which was about a year and a half after the idea was created. I was involved very much in the development of the first website that helped enable this. And we started first with phenotypic drug discovery. Reason for that is, in pharma, a lot of concern about disclosing what are your priority targets, where are you going after. So with phenotypic, most people know what therapeutic areas we're working in, where our interests are. As we had success in that, and as most people know, most of the pharma or otherwise are operating in similar target space. What we came back and really 2 years later with the revamped website, opened it up to actually do the analysis relative to the specific targets. A lot of challenges in terms of this as we set this up, because now we're actually in this thing is reaching out to individuals and asking, do you have compounds you want to screen in our assays? We'll give you them information back, biological data, use it as you will. If we see something of interest, we'd like to potentially collaborate with you. Real concerns around trust, IP ownership, how do you transfer the materials back and forth, MTAs, especially if we're starting to do it from countries like Russia, where I don't think we actually have an MTA set up yet, or other ones, and now you have to transfer these compounds back and forth around the globe. The website, how do you make it efficient? How do we make it incremental? How do we not make it operationally complex? And then also, now we've become, in some respects, a service provider. We're providing data back. How do we do that with quality and ability? So these were all things we had to do and as we went forward. And the way we really thought about doing this was, again, in a web-facilitated way, to really do this where ideas compounds, structures were submitted to the website. They would actually go through our informatics tools. We didn't know how many people would start submitting structures. We didn't know how different they would be from our collection. We didn't know if there were things we didn't want or not. So we actually had them upload. We had ways to do analysis on them, and then an ability to, if they passed our initial filters, to allow the scientists say, yes, I have this sample, I want to submit it to you to do it. And then we had to get into all the operational specifics of them actually putting compound in a vial, shipping it back to us putting it through assays, and in the end, delivering a report back to them. And all of this, and I'll talk about this, is without Lilly ever seeing the structure. What we patent and what we do is small molecules. We did not want to see the structure in any way be contaminated by the structure relative to our project teams. And so all of this was generating it without seeing that. And in the end, if we saw something of biological interest, then we would reveal the structure. And I'll move through these really quickly, but again, as we go, whether it's academics, biotech, small companies, they'd submit the compounds and ideas to us. Eventually, Lilly would use our assays and in silico models to evaluate those decision-making, and in the end, data goes back to the investigator who we're collaborating with, and if there's something interesting there, then we will actually reach out to them, ask, can we see the structure, so that we know if it's something we might be interested in, and start a collaboration as such. If you go back, originally we started with the phenotypic assays that are here, and all of this is on the website. So the website actually has a huge amount of this information, more than what I'll actually present here. We then moved into various targets, which were predominantly endocrine, oncology, and some neuroscience. And we also have a key effort in tuberculosis, MDR-TB, and also now starting to say they could opt in to having their compound screened in the TB assays. So let me move relatively quickly here. I'll give just a comment or two around this. One of the things we, again, want to be very careful about is we put the database and the structures out in outside our firewall, so that we would not be contaminated by any of these. However, we also had some cheminformatics tools we wanted to leverage internally. So what we did was actually use some generalized fingerprints to encode the compounds, only use the generalized fingerprints to transfer across the firewall to run on our systems, to do some predictive modeling, some similarities, and then give the results back out, and then remove the internal cache. We've been very careful not to try and mine any of the information that we have here without the investigators' information. So this was a critical thing in terms of how we set this up. As we then went forward, this is the new designed Open Innovation Drug Discovery. This is the second version now, where we actually handle most all of those. If you just do a search of Lilly Open Innovation, this should be close to the top one there. Lots of information you can get to and how we do the informatics, how we do the various things. What I want to walk you through just a little bit in terms of what it looks like, standard logins. Once you have an account set up, a variety of ways for people to upload, again with ChemAxon tools, ability to upload libraries, compounds, and then submit them for processing. As they go forward, once they've been processed, they can actually then, and they have a variety of different table views in terms there. Once they've been processed by informatics, they can submit them. Then there'll actually be some capabilities that if they're submitted and the administrator actually accepts them for samples, then there's abilities for, we will actually ship out boxes prepaid for them to put vials in. When they do put them in the vials, then they come to the site and put the barcodes in, so we know what we're receiving as we go forward, as well as what's there. And then ultimately, once it gets on our side, they're registered, solubilized, put through the assays, results generated, and given back to the website here. So We went into this not knowing what the uptake would be, what type of people would be involved. And at this point, the most recent data is we have 280 affiliations. Our agreements for MTAs are with the affiliation, and then the professors, the scientists, the researchers then become a part of that affiliation and can submit under the MTA to us. So you can see them spread across the globe, and you can see how it's increased as we've gone throughout the launch of this site. So very good uptake on this. But the question becomes back to, well, how much is being submitted? How much is passing our filters? So if you actually look at this, we have a huge number actually that have been compounds that pass the informatics filters. It's roughly about 50% pass the informatics filters. They're looking for things, medicinal chemistry rules. Are they the right type of compounds? Are they controlled substances? Are they too close to Lilly? Because we don't want to actually screen things that are very close to us. We have them in our collection. Or are there other, you saw Lipinski filters, you saw solubility filters that was shown here. We have some of those internally, and so we have a desirability score we also use in terms of, does this really look like it's something we're interested in? Huge number actually that have passed. The numbers are being cropped a little bit on there, but 22,000 that samples that actually received, 17,000 that we have gone through our assays. Huge amount. So the question becomes back, well, are we gaining anything from this? Are they more different from what our normal compound collection was? And we were very pleased to see that actually, if you look at this is a Tanimoto similarity score, you actually see a large amount of them are not only different from the Lilly collection, so we're seeing something very different than we might have seen from our own scientists, our own compound collection. In addition, we're also seeing them different than PubChem. What we were also worried about, would a lot of people just be submitting PubChem compounds to us, and did we just become a screening center for this? Well, no, we see quite a bit of variability. And then the question becomes, well, but are they biologically relevant to what we want to see? Or are they all just brick dust? Are they all just sitting there? If you actually look at the hit rates then, in terms of these various ones, where you see in green are the phenotypic and the yellow are the target-based, you actually see very reasonable hit rates in terms of these across our collection, not much different than what you'd see from internal to a pharmaceutical. And the project teams are actually running these projects as they go forward internally. And so we've accessed now different compounds, different types of chemistry, different types of biologically active as we go forward. So then the third thing is, we're not so worried about all these type of things. What we really want to get to is really collaborations. Did we find something that's interesting for us to work on and move forward? Because we don't have collaboration, that's the whole goal of this. And so what you actually will see is what we're looking for in collaborations is we're looking for various things. Is there an opportunity for someone to bring some innovation uniquely to us of different, where we could fund the research, where we could move them from an idea or compound to a lead or to a potential future candidate in these areas? Can we actually not Lilly only just do all the SAR, but can we actually use the academic lab to do the SAR themselves? And so we can jointly develop something going forward. And so we have a variety of mechanisms in terms of this to how do you progress this going forward. And so these are, and on the site they have some of the more recent ones. The ironic thing about this, when you look at it, is we look to access innovation around the world. Our first collaboration came like 2 hours up the road from Notre Dame. Just how it happened, but then if you look at the other collaborations from China, France, Canada, really across. The other key thing that this has done to us, or done for us, is really to give us access to press and collaborations that we wouldn't have had access to before, because now people were coming to us with new ideas. The websites and the tools that we delivered actually were part of a Information Week Business Value Innovator Award last year. We're getting a lot of press in terms of that's opening up doors for new collaborations as we go forward. And even with the NIH, where we're now coming back in terms of screening various compounds and doing this. Now the data generated, it goes back to the people submitting the compounds. It's up to them what they do with them. They can publish, and we have all of the references of the assays and everything online so that they can publish that. So wrapping up now here, what's next? In the near term, we actually want to do some more SAR and SPR models in the interest of not just generating data, but collaborations and people who can learn to drive medicinal chemistry. We want to have those tools available to them. We're actually looking at ChemAxon to help us deliver some of those tools going forward, because we're not a software development company. We have some good tools inside, but we ultimately in the future don't want to have to support all of those things. Improved reporting tools, we want to make it easier to get the samples in as opposed to dry powder. Improve into new types. We started with small molecules. We've actually now accept natural products. We also have just opened it up as of the middle of this year to peptides going forward, and then we will continue to move up. There are challenges as you continue to move up the scale in terms of how we accept those compounds for screening. And then long term, we really actually want to access innovation not only in the small molecule space, but in the disease space, in the assay space, or anywhere else where someone has a novel capability that may be useful for us to tackle the next versions of disease as we go forward. So new targets, new assays, any of those ideas, and ultimately in the end, continue to assist, just like in the IT open source community, continue to be a viable partner in the broader network of open innovation as we go forward. And at that point, I'll again just key wrap up in this. This is one of our critical projects as we've gone forward. ChemAxon, both in the past and we expect as we go forward to continue to be a player in this space for tools of choice as we go. So at this point, if there's additional questions.
It's very innovative, actually.
Yeah.
Are other pharma companies doing things like this?
So we have looked at those various ways of tackling the problem, and what I will comment to this is when our VP at the time, Alan Palkowitz, came to us and said, this is what we wanted to do, Lilly Midwest, I said, you got to be kidding, right? I mean, we're so conservative, how would we pull this off? Because it really was a very different paradigm from what we did with security. Yes, there's various ways tackling the various ones. I can't speak for all the companies, but various companies are setting up research centers, they're setting up research sites to allow innovation. We're all tackling it various different ways to say, how do we get those collaborations? How do we reach beyond our walls and do this? I know there's other people who are interested in this paradigm where we're actually pulling things internal to ours, assaying them, giving them. We haven't seen anything quite like that yet, but we have seen lots of things relative to researchers, setting up sites, postdocs, other type ways of doing this. And Lilly likewise is continuing to do other models from this in terms of open innovation and setting up that collaborative network going forward. Different than the outsourcing or some of the other shared risks that we're doing, really trying to help the broader community.
So the molecules get submitted online, more or less, right?
Yes.
And then it's basically just a structure at that point before
Correct.
you decide to get and you must have an extensive computer workflow, so to speak.
Yes.
In order to determine its eligibility.
Yes, and in fact, if you actually go to the website and look under evaluating compounds, you'll see a white paper there where we talk in general about the MedChem rules, looking for substructures we don't want to see, similarity to various compounds, not being similar to controlled substances that we know about. We have some predictive models that we use. We don't disclose all of that, but we get relatively close to what the workflow is in terms of doing that. One key thing when we actually went, and originally we thought we'd get inundated by amount of compounds, people submitting, we'd be generating too much data, we can't, it's competing with our internal assays and internal data. What actually happened about, I think it was 2.5 years ago, I was sitting there with a couple others, the person running the screening, the person running the project, and yes, we were filtering quite a bit of compounds down by human intervention, by the desirability score. What we actually saw was, we weren't seeing too many people that were submitting more than we could handle. We actually started anything that was accepting passing the cheminformatics filters. We were almost automatically accepting them unless it was one individual or institution that's sending, 10,000 compounds,
yeah.
right? But most of them, we actually just opened up the flow and let the data be generated.
It's very nice.
Yeah.